Hi, Lily.
Hi, Charlie. What are you working on?
Microbes.
And why do you study microbes, Charlie?
Why'd you ask?
Because I'm studying scientists to find out why they do what they do.
Well, it's a good thing to do. What is there to study?
Whether science is a good thing to do or not depends on your values.
Values?
Yep. They're what motivates you and tells you what's right and wrong, what's a good project and what's not. We need to know what values are shaping big projects like the COE.
Okay. So what are our centre's values?
Well, I've been asking COE scientists why they do what they do. Take a look.
I do science to promote well-being and solve problems.
I do science to discover new knowledge.
I do science to avoid harm.
Another values question is, how do you do good science?
Good science is objective. Good science is rigorous.
And reproducible. And then a third really important question is, how do you be a good scientist?
You need passion, creativity, and curiosity.
You need humility and honesty.
You need to be collaborative, but there also needs to be good communication and leadership.
Oh, you also need the resilience and determination to hang on in there. We worked out 3 categories for values: motives, personal, and scientific, and found factors that support or undermine them.
That sounds like my job. Working out what makes microbes thrive or not do so well.
Exactly.
Good afternoon, everyone. Meet the COE Values Microbiome. We've identified 3 main genera: Pseudomonas motivatis, Helicobacter scientifica, and Saccharomyces characteri. There are 3 main forms of Pseudomonas motivatis: doing good, avoiding harm, and discovery. All 3 forms need funding to survive. Funding comes from government grants or industry collaborations. The mix makes a difference to which ones flourish. The doing good Pseudomonas adapt to lots of industry collaboration by evolving into a new problem-solving form. But the discovery form can't metabolize too much industry funding and may wither or die without government support. Helicobacter scientifica are quite sensitive. They live in mucus layers to protect them from various threats. H. scientifica have 2 forms: rigor and objectivity. These need a supportive environment to flourish. The most diverse genera in the COE Values microbiome are Saccharomyces characteri. There are 5 forms. The Saccharomyces are key to a healthy microbiome, but they are susceptible to bias and power imbalances. The Values microbiome is in constant flux, interacting with external factors and each other. In our next study, we want to sample the entire COE for unknown species, traits, and interactions. Any questions?
Don't we already know all this stuff? What's the practical benefit of your research?
So, yeah, what is the point of all this values research?
Well, achieving the goals of the COE needs a specific set of values. Before we started our research, we didn't know what COE members' values were. And now we do. We can explore how these can help us reach the COE's goals.
Ah, it's like you can't get to your destination without knowing where you are.
Exactly. So, can I interview you now?
Sure.